Okay, how did people like the paper?
Straightforward.
Yeah, pretty.
Yeah, I liked it because I could understand it.
Oh, that's good. All right, so I don't think we need much background for this one, right? It follows the stream of papers that we've been looking at. It's more in the taste system. Yes, by now we've heard a lot about PBN, so we can jump just straight into the figure one. Yeah.
That's me. Okay.
Okay.
So what they did was they introduced AAC into Cre-dependent M-cherry cells to confirm the selective expression of M-cherry. So that's A. And then for B, they injected Cre-dependent diphtheria toxin chain, or DT. They kept calling it DTA, in order to ablate and lose those SATB2 PBN neurons. So in B and C, you can see that this was successful. So then what they did was they counted the number of licks in a short amount of time to measure taste sensitivity. And D showed that mice without the SATB2 neurons had impaired sweet taste. And E through H shows that the other taste remained intact even without the SATB2 neurons. So it looks like these neurons are important for sweet taste sensing.
That's it. So simple.
I know. I got a good one.
All right. So, what are they staining for in A and B? What are those green cells?
Those are the SATB2 positive. Can you hear me?
And,
Okay.
And through how are they labeling the SATB2 neurons?
Cre-M-cherry.
M-cherry will be red though, right?
Oh.
It's an immunostaining, right?
With SATB2 antibody?
Mm-hmm.
Look like so.
So then they either inject DTA or not. Sorry, I'm trying to pull up the paper on my iPad. So they have a SATB2 Cre animal, or they have SATB2 Cre animals. And then in some of them, they inject M-cherry so that they go through the surgery, the anesthesia, open up the scalp, drill a hole in the skull, and then inject M-cherry. And in some animals, they're injecting the AAV Cre-dependent diphtheria toxin to kill the cells that express Cre. And after that, they're injecting, or not injecting, they're sacrificing the animal, sectioning to get the PBN, and then using a SATB2 antibody to see how much SATB2 remains. SATB2 expressing neurons are still there, right? So that's A and B, and C. So C just counts the number of cells. I mean, I gotta say that I don't know how big a region they counted the cells in, but as opposed to what you see in A and B, which seems like you're getting from 1,000 cells to 50 cells, their graph actually isn't that dramatic, right? It's like a 50% reduction, right?
So are the most region just because they exclude the region?
Exclude which region?
I mean, like for A, if we see the PBN region they draw, it actually, most of the cells labeled green are not included, right?
Oh no, the dotted line is the peduncle. It's where
Oh,
it's the area that's connected to the cerebellum, right?
oh, so the region below that is
Yeah.
considered.
So then this is what people call. Jahao is an expert in this region. Right? It's where people call like the medial region and the lateral region, the dorsolateral, the external lateral. This all predicates on. It's related. The landmark there is the peduncle.
Okay. Oh, okay.
So that's what the dotted line is. The superior. How do I remove this? Yeah, it's the superior cerebellar peduncle. That's dotted line. They probably flushed the entire area with the diphtheria toxin virus, right? Don't you agree that that from the images it seems like the DTA worked amazingly well, but in fact from the graph it doesn't seem like that's really the case. I don't know why.
Well, they're showing only the best pictures, so they want to show us that it kind of worked, but then the graph tells us it's only 50%.
But in any case, they're getting about half of the SATB2 cells to die, and because of it, maybe not send up information into the brain that sweet is good. And the method that they assess this is by seeing how many licks animals take to increasing concentrations of a tasteless, right? And as Amanda very nicely explained to us, the effect is in sucrose, but other tastings don't seem to be affected by it. Yep, pretty straightforward, right? But everyone understands why they're using mCherry as control and DTA as the experimental, right? So I have a question about how this is done. Why would the F animal in F, right? It at almost very low concentration of denatonium, which is bitter. They're licking at this point. They're licking about 70 licks per 10 seconds, which is about the maximal speed that the tongue can move in a mouse. Why are they licking this, but not licking 10 millimolar sucrose? Wouldn't you expect that to be like maximal level? Why would that be?
Is the bitter response just like significantly stronger than the sweet? Because in figure 2 later, I noticed how for like the bitter, sour, and salt, there was a gradual increase in activity compared to sucrose, where it was like very small. So maybe it has something to do with like how intense the neurons respond, maybe.
But But if the animals are willing to lick slightly bitter substances, right, in F here, why wouldn't they lick slightly sweet substances here? Yeah. And Alex, having done some of these lick assays, might be able to tell you that these behaviors are not straightforward, right? So how do they get animals to lick denatonium? Why would they even lick something that's bitter?
Are they also water deprived in this essay?
Of course they're water deprived.
Ta-da!
There's
So
strong,
they see a spout.
their strong motivation is to be not thirsty.
Yes.
And if I remember correctly, they played a slight trick where for bitter, they water deprive or water restricted more longer, maybe like 20 some hours. For sucrose, they're water depriving less so that at least the mouse would go to the spout, but not really feel very motivated to do so. So their motivation for the sucrose is that they like the rewarding aspect of sweet. The motivation for these mice in F is to be not thirsty. But at some point it becomes just too bitter, and at maybe like 1 millimolar denatonium, the bitterness wins over the thirst, right? And they stop drinking.
So they train the mice and then they deprive for 4 hours for appetitive taste and 23 hours for aversive tastes.
Mm-hmm.
So that's a huge difference.
Mm-hmm. And that's why at close to water concentrations, they're not, these mice are not licking, but these mice are licking.
That makes sense.
And what else? This glutamate effect is something that's actually quite hard to get. The umami taste, it doesn't really happen that easily. So it's nice that they see some umami responses. It's probably why they don't see much until 100 millimolar glutamate. So one of the main differences in our taste system versus mice is that when the umami receptor was cloned, this is T1R1, and the co-receptor is T1R3. So when T1R1 was cloned, they put that T1R1 into a cultured cell line, and then they expressed it in a cultured cell line, and they added various amino acids onto the culture medium to see which cell, if the cells responded to the various stimuli. And they saw that with the mouse version, the T1R1 binds quite well to all 20 amino acids. But the human version had much higher affinity to glutamate as opposed to other 19 amino acids, which is why we love MSG. So I think we can move on to the next figure.
I could take this one because I have a few questions on it.
Okay.
Okay, so in A, they're showing the surgical schematic of where they injected this AAV human synapsin flex DCAMP S virus.
Mhm.
So the idea behind this figure is that they want to test or evaluate the responses in vivo using a DCAMP, a functional way to study how these cells respond. And so they injected this DCAMP-S into V-Glut iris Cree mice, which I was very confused about because I just don't understand the motivation for using the V-Glut versus just the SAD-B Cree mice that they already had if they were testing the function of the SAD-B neurons in the first place.
Mhm.
So that I was confused about. And to the right of A, they're just showing the expression of this DCAMP6S and the SAT-B2 neurons. So you can see that after injection, there are DCAMP-S positive cells. And D through G is another, they're showing the delta F over F, so the difference, the increase in fluorescence upon exposure to water, sucrose, glutamate, umami paste, denatonium. I think that one's bitter, and then citric acid and NaCl. And what they're showing in, I guess the colored schemes are the different tastes, and they're just showing that upon all of the tastes, upon exposure to all the tastes, there's an increase in that delta F over F as compared to water, which is the under sort of, I don't think you can see my cursor. Under, it's like the shaded area under the curve kind of thing. And then in H, they're just quantifying the delta F over F at the different taste qualities. And there's a significant difference compared to water. But yes, back to my questions. I was just wondering about why did they use the flex? Also, you think why did they use the flex AAV virus to express DCAMP-S on V-Glut iris Cree mice? I was very confused.
And did somebody want to answer that? Why would they look for, why would they not go straight into SATB2, but look at just broader responses? Any suggestions?
I mean, I understood the part where they said that there is a high proportion of V-Glut positive neurons in the waist area of the PBN. So that makes sense. But why not target specifically the SAD-B2 neurons? That's like part of the question. And then
Well, let's just think about assuming that SATB2 is encoding sweet, right? If this exact experiment was done in SATB2 Cree animals, then what would you expect to see? Would you see anything with water?
probably not.
Okay, so would you see something here?
Sucrose, yes.
Right?
Yes.
Here? Probably not, right?
But they were just like on the first figure, they were just showing that upon exposure to glutamate at 1,000 millimolar, they do see it, like an increased lick response. So that's why I'm like, you do see.
But what they're seeing here is that animals like glutamate, right? But even if you kill SATB2 cells, animals still like glutamate.
Oh, so these are the D through F here are the ablated, like post-ablation.
Yeah.
Okay, that
So
makes
you're
more
using
sense.
diphtheria
Oh,
toxin
okay, that makes more sense. I was like, there's no way, this makes no sense. Okay, now it all makes sense.
Okay. So
Um,
then from figure 1, what they're seeing is that when you kill SATB2 neurons in the PBN, you're affecting sweet but nothing else. So
okay,
presumably they're conveying sweet information, sweet taste information.
that was the takeaway of
Yeah.
figure 1.
So-
Okay,
So then they could go straight into SATB2 Cree animals and do this phybophotometry experiment, right? And measure neuronal activity in bulk by presenting different taste tints on the tongue, and I would assume that you would see a response with sucrose, but nothing else, right? But that could, why would that be a problem? What would that not show us? I mean, in the figures that that will come later, they're all focused on the SATB2, right? So I guess the reason for doing this in a broader collection of cells is because you want to show that you're in the right position where taste information goes into. Let's say you did this experiment and you just saw sucrose response, but to nothing else in this VGLU2 animals, then you'd be concerned that maybe it's this topologically, you're getting sweet responses in this region, but nowhere else, nothing else, no other taste, right? So it could point to several other ways of this working, but just by showing that in this general vicinity, you're getting activity in excitatory neurons, which is what VGLU2 is. You're seeing activity of taste information coming through to sweet, umami, bitter, sour, and salty. Makes you think that this general area is where all the taste information passes through, right?
why did they use the flex Gcamp versus a normal Gcamp, I guess?
As opposed to just doing synapsin Gcamp
Gcamp,
without the
yeah.
Cree.
Mm-hmm.
They could have, they might have, but maybe they see a better result just limiting that to excitatory neurons. Perhaps there's an effect of inhibitory neurons that will make this analysis messier. I don't have a good answer to that, but all I can say is that with the VGLU2, they're seeing a pretty robust response, right? So this is, Jaha, would you say this is a robust response? It's like 2% or like 4%, 10% at most, right?
I think compared to the trace itself,
Yeah.
I think so. The trace itself is really much smaller. I mean, the thickness of this line itself is much smaller. So comparatively, yes.
Yeah.
So I would say probably they're not averaging multiple across trials. So if you are averaging, you should see a variance of those peaks, let's say.
Yeah.
So you have at least you have like a variations.
Mm-hmm. So this is probably a representative trace, right? Yeah. Because they also want to show with these peaks, spikes, the licking, right? So there's, and the purpose of that is they want to show that the activity of VGLU2 neurons in this region is tied to the lick or the tasting of the stimuli, right? And I think they keep on harping on the point that this happens only after the lick is taken. So they're not forming an idea of what the stimulus might be, and then that expectation showing up in the PBN. They don't really see that. And that's a valid point, because even though the spout might look the same, maybe the animal can smell what the stimulus is, and in that case, the spikes might precede the licking. Right? Just when the spout opens, the shutter opens, the spike, the spout is there. Maybe they could see spiking in this region. If they were to look in the insula, right, in the gustatory cortex, we've seen cases where the animals are aware of the context and they respond to that as opposed to a taste response. But this seems to be a purely taste response. Okay. So Jiahao, is this, I guess they don't really specify which region they're measuring from, but I would expect they're going more medial, right?
You mean which area for the figure 2?
from wherever they're looking at. I don't know if they're actually distinguishing which region of the PBN they're looking at.
First of all, I think Alex made a very good point that shows the best picture.
Yeah.
And second is specific to figure 2. Figure 2 actually not only expressed in the medial part of PBN.
Yeah.
They're everywhere.
Mm-hmm.
So it's nice that they show us this. I hope what they show in the figure 2A, the immuno where the major GCAM signal is situated in the medial part, all the rest of the signal are coming from these mass. Mm-hmm. Then I'm fine. Then that's fine. I hope they are, let's say, corresponding to each other.
So when you did the P2X2 Cree GQ stimulation, and then you saw a lot of staining in the PBN, would you say that this region in figure 1A is a big part of where you saw the activity?
No.
No?
No. Don't feel so.
Okay.
A rather lateral part.
Yeah.
And natural dorsal part, kind of.
So like here?
Yeah.
Yeah. Okay. And here is still more medial, right?
This is much caudal for PBN.
Yeah.
And this is actually where most of the taste area situated. Mm-hmm. So.
All right. So what do they do in figure 3?
Then now zoom in and say, okay, do the same techniques, inject the virus, express the GCAM in the SAP-B2 positive cells, and then insert the fiber and see what happens when the animal licks different tastes. Indeed, show us before licks, the first sensor is high, so that's represent the baseline. So once they start leaking sucrose, they got responses. And the higher sucrose is 500 mils, you got larger GCAM response. And then you have sucralose, which is, I think is chemically similar to sucrose. but it's not taste sweet, or it's taste sweet.
No, it tastes sweet. It's an artificial sweet.
Not caloric.
Yeah.
So it can be metabolized by the animal, but still tastes sweet.
Yeah.
So the sweet itself, so it means like sweet is those cells, SAP-B2 cells, responds to sweetness itself.
Mm-hmm.
And then all the rest, even glutamate, if you remember in the previous figure, glutamate, denatatolamine, and all the other tastants actually evoke the PBN responses in the excitatory cells, but they are not evoke any responses in the SAP-B2 neurons. Mm-hmm. So overall, this indicating a specific responses of SAP-B2 towards the sweetness.
Yeah. And
then the author just pick out two extreme cases comparing the sweetness versus the bitter by applying the 500 millimolar sucrose versus 10 millimolar denatatolamine and see how this goes. And this is actually let's say average trace for fluorescence. Even from here, I would say it's pretty good. The single noise ratio is pretty good. Yeah. It's just like the imaging itself is a bit horrible. I cannot believe it. If they are the best, if they present us the best case.
Yeah.
And then I think they are using a different techniques with the endominiscope, so they can record calcium signal from individual cells. So they're using the putmasing the same behavior apparatus, and right now we have more resolution on cellular level, single cell level. So still denatatolamine doesn't give much responses until like about 5 seconds later. And then for the sucrose, you have a lot of neurons. Like I think from here, maybe 1/3 or more, a little bit more, of neurons that responds immediately when the sucrose, when the animal take in the sucrose. And there are some other cells have a delayed response, but it's overall majority of those recorded neurons, or I'm not sure whether those are the only cells that respond. Let's say whether they present us all the neurons that recorded by their miniscope, or whether those are the ones that show some response. Those could be lead to different conclusion. But overall, they saw the cellular responses specific for preferentially for the sucrose stimuli. Mm-hmm. So that's all consistent. Yep. This was a hypothesis that SAP-B2 neurons preferentially detects or responds to sweetness.
Yeah. And I don't know what these three cells are. They don't seem to be responding to sucrose. But I don't know if they're even responding to bitter. And so, but you do see a more detailed picture of what's going on in the individual cells here, in the 30 cells that they can see. Yeah. Is that clear for everyone? Yeah. So everything's making sense? Yeah. All right, so just to recap, they identified this new population of neurons in the PBN, SAP-B2, which they think is necessary for sweet taste detection, because when they killed with DTA, they see that these animals are less attracted to sugar. They also see SAP-B2 neurons responding whenever animals are licking or detecting sugars, sweet. So they want to do functional studies, right? So that's what figure 4 is. Who's going to go over figure 4 for us? Nobody?
I can do that.
Okay.
So in panel A, this is just a demonstration of how they did the experiment. So they injected the channelodopsin, create-dependent channelodopsin into the PBN region, so that not only the cell bodies of those PBN neurons will be labeled, as well as the fibers that is extending to the 3 regions, VPM, central amygdala, and BNST. They are also labeled green or yellow. So B is just a YFP expression that indicates successfully expression of the channelodopsin. So in C, they are trying to use optogenetic to activate these neurons, so that we can see there are 4 in total. The PBN, it just indicates they use the optogenetic activation that's directly on the neurons in the PBN. And for the other 3, they are activating actually the fiber terminal that are extended from the PBN neurons, but presynaptic to the neurons in respective region. So for example, if it's VPM PC, then it is activating the fibers extended from PBN, but right like after they're activating the neuron, before they're activating neurons in the VPM. So here we see that only when so if they directly activate the neurons in PBN, like in the first column in panel C, definitely there is a very strong increase in the leaking frequency of these mice, indicating it's mimicking they are getting a very like appetitive or rewarding feeling.
So in that experiment, they're injecting channelodopsin into the PBN, right?
Mm-hmm.
And shining light in the PBN, in the cell bodies, right?
Right. So that further confirms PBN is related to a rewarding, like
yeah. And does everyone understand how this is done? They're presenting animals with water, a spout with water, right?
Yes.
And at times the light is shown onto the PBN neurons, and at other times light is turned off. And you can see that when the light is on, they tend to lick more than when the light is off, right? So and by light I mean that they're activating the SATB2 neurons in the PBN.
Yes.
Yeah.
So similarly, in the VPM region, they have a similar result. As the mice start licking, they shine the light, and the mice will increase their licking frequency. So indicating they are feeling they are tasting something sweet or like a rewarding. But for the amygdala and NSD region, there is no significant difference. So also they did I think that's a preference test, right? So indeed, they are just also they have one side. I think they have the mice freely moving, and when they're reaching one side, they have the light on the other side, they have light off to see if the mice have a preference on staying one side. And similar result here in the VPM, when they're activating the neural terminals in the VPM, the mice definitely would prefer to stay on the side where there's light staying on. But for the amygdala, there's just no significant difference.
Mm-hmm.
And E is just a quantitative representation. So overall, it is consistent with the taste, like a processing pathway where beyond the PBN, the signals are mostly transmitted to the VPM region and further to the gustatory cortex. So here they're just confirming that and indicating, also facilitating their conclusion that they are finding part of the sweet signal processing pathway.
Which on one hand must be very reassuring, because from the PBN to the VPM, this is a very well established taste pathway. So generally people think that it goes from the tongue, ganglia, NTS, PBN, VPM, and all they're seeing is that main taste pathway is conserved, and there is information going from the SAPB2 neurons in the PBN into the VPM, because they're not even looking at the next neurons. They're just looking at the projection of the SAPB2 neurons into the various regions, right? They're not, it's not like they're seeing sweep neurons in the VPM. All they're doing is they're shining light onto the terminals of the SAPB2 neurons. So presumably there are neurons that take up the postsynaptic neurons that in the VPM that get information from the SAPB2 neurons, but they don't know what it is. So yeah. Go, you were saying something?
I mean, I think it's very like reasonable if they want, they can do like anterograde tracing to see how these signals are processed, right? Instead of just getting one step beyond.
You mean like a rabies kind of experiment?
Yeah, or HSV. I mean, then they can definitely see a clear pathway, right? Beyond VPM, even they can see where these signals are projected to.
Yeah. And I say that this is reassuring that the established pathway is correct, that they can confirm the established pathways. But on the other hand, they don't see much with other regions where they see projections of the SAPB2 neurons go to. They don't see effects in the amygdala or the PNST. So that must be slightly disappointing for them, right? Because if they were able to say that, oh yes, the main taste information goes up to the gustatory cortex through the VPM, but there's a side branch that goes to the amygdala and it confers immediate valence information that might have looked more interesting in the grand scheme of things, but that doesn't seem to be the case. Right. I think overall this paper is quite straightforward. So we're able to go through it in less than 50 minutes.
I just have a question. To start a project like this, like how do they initially start to thinking about SAPB2 might be a candidate for taste system?
I think they must have looked in the Allen Brain Atlas, and they said they looked in this region where taste information is supposed to go through, right? Because if you look in, let's see if I can pull it up. So if you look in the supplemental figures, this is the expression, so not necessary. Here, this is quite interesting because they look at the excitatory neurons with V-GLUT versus the inhibitory neurons, and they think that most of the SAPB2 neurons are excitatory, which is why they did this. They used this in figure 2. And then they must have done these kind of screens where they present sweet or they present bitter, and then do C-Fos stains and they saw that many of the SAPB2 neurons were activated when sweet was presented. Yeah. So my prediction is that they looked in the Allen Brain Atlas to see what potential markers there might be in this region, and then did some kind of a screen with CFOS and settled on SAPB2. I think that's it. Yeah.
Okay.
I wonder how they did the taste and presentation here. Because you were doing this kind of a CFOS staining in the NTS, right? It might be worth looking into how they presented these various tastings, whether this was in awake animals, sleeping animals, if they force-fed, how long they presented various tastings for. So, because we might be going back to this at some point.
That's right. Let's see.
Questions or comments? No? We're all good? Too easy, too straightforward?
I have a question about figure 3
Yeah.
and D.
Figure 3B.
D.
Yes. Yeah.
Yeah. When tasting the bitter things, after five seconds, some cells are activity.
Yeah.
Why they are active to bitter taste?
Who knows? And I don't even know if it's specific to bitter.
But we see the same activity in D after 5 seconds. Suddenly there's more, like 10 more neurons activated.
Here.
Yes.
Yeah. Actually, interestingly, there's activity here also. In C, right? Slight, but there's something happening. Yeah, I can't answer that. I don't know why. It might be something interesting because these neurons do respond specifically to sucrose, right? And then towards the end, when they're ramping down, there's some activity to denatonium.
The metallic aftertaste of the spout. I don't know, maybe there's something there, but probably no idea. No idea.
It's interesting. It happens much later. I don't know what it is. Yeah, it's not like it. So in this animal, they're presenting denatonium, and then for 5 seconds nothing happens, and then a few sweet neurons light up. Yeah, much more interesting than cell number 1, 2, and 3, where they just seem to be non-specifically active, unrelated to licking. Right?
But I don't feel they want to, let's say, dig a little bit into these cellular responses in SAPB2 neurons. Probably, so I even don't know how to interpret their calcium results, because they are not telling, I'm not sure they're telling me whether it's the first encounters leaking or the not leaking, or whether their interview, let's say, no, they randomize the denatonium versus the sucrose presentation, or how they do that. I feel somehow there's some tricks there.
Actually, that's a good point. Do you think they mixed in sweet and bitter?
I don't know, because it's like two, let's say, at least 3, let's say, maybe water also there. So it's a bit, if there's 3 atoms, I'm not sure how much randomness there are. And second is those 30 units coming from 3 animals. 3 animals is barely enough for function study. It should go more.
Yeah.
And then, but I don't. That's one of the reasons I don't think we need to dig in too much of this, speculate too much, because we don't have enough
agree.
things to. Yeah.
And the fact that that there's a sharp line where denatonium seem to stimulate these neurons.
Yeah, and sucrose not.
Yeah, this must be more of a maybe an engineering thing, or I can't see why there's a sharp line here. Maybe it comes from the same animal, right? And
Oh, those are definitely from same cells.
Yeah.
So the number, each of the cell number, for example, the number 10 in denatonium case is the same cell in the sucrose case.
Oh no. What I mean is these 7 cells are probably from the same animal and same recording.
Oh.
Yeah.
Who knows?
I don't think this happens in like a few from mouse number 1, a few from mouse number 2, a few from mouse number 3. I would expect this all happened in one animal, so that there might have been something that happened during that time.
And also with miniscope, 10 cells each mice is a little bit small. It should have like hundreds of, maybe at least around the hundreds of cells for each mice. So no matter whether they respond to sucrose or not, it should be included into your analysis. So, but very good point that to show like the different interesting thing happens when we look at the grouped calcium signal versus the individual cellular calcium signal. So there will be definitely more interesting thing when you look at the cellular responses.
Yeah, single cell resolution. This is why you do single cell resolution. And this is why we have the 3-photon to show responses in the MPS, right? Okay. Unless there are other questions, I think we're done. Yeah.
Great.
Okay. Well, enjoy the cold weekend. Is it going to be nicer?
Not maybe.
No? Somebody said
No.
It's going to be raining for the whole week next week, or at least like sunny days.
All right. Well,
October, yay! Have an umbrella with you at all times.
I remember how much it snowed in Halloween last year, so.
Yeah.
Okay, so Monday, Kyujin will be joining us freshly out of quarantine. So we'll have one more person in the lab.
Yay.
That's nice.
Okay. All right.
But it will be very windy tomorrow.
Jahal, I was trying to end on a positive.
That's not a positive thing.
Being very windy and cold and nasty.
Yes, this way you can get the fall, all the leaves into your face. You don't need to look at them. They come to you.
Yeah. That's right.
Maybe I need a goggle.
All right. I'll see you all in a couple minutes.
Yep.
For people remote. But thank you. Have a nice weekend.
Have a nice weekend.
A nice
See ya.
weekend.